Oi, pessoal. Antes de eu passar a palavra, né, para o Magno e para o Andrej, falar algumas palavrinhas para vocês, como a gente sempre fala, né? Primeiramente, antes que a gente esqueça, a gente tem demais que agradecer a Analítica pelo apoio que ela tem dado para a gente. Sem ela não seria possível ter essa estrutura que a gente tem ultimamente. Então, muito obrigado. Lembrar também, né, que quem fez a inscrição no evento, a lista de presença será colocada no final dessa sessão, tá bom? Vamos colocar aí também o link do nosso WhatsApp para quem tiver interesse de entrar, eu aconselho, bem legal. A gente discute muita coisa lá. Que mais? Eu gostaria também de falar, a gente teve, né, a próxima sessão, semana que vem, vai ser a sessão dos estudantes, e eles já foram selecionados, então, já foram também comunicados, né, por e-mail, pela Ana Carolina, que é uma das organizadoras do evento, mas eu gostaria aqui, então, de dar os parabéns, lembrando que essa seleção foi igual a gente fez para o anterior, então são pessoas que são da diretoria e do comitê, esqueci o nome do nosso comitê, do BrProt, né, do conselho, que fez essa seleção. Então foram 9 pessoas. É importante falar que, por exemplo, se eu tivesse participando de algum trabalho, eu não participava dessa seleção, daquela avaliação específica, isso é importante falar. Tá? Então, nós temos 6 selecionados. Então, a gente gostaria então, de dar os parabéns para a Janaína Macedo da Silva, a Jéssica de Siqueira Guedes, Lícia Silva Costa, Lucas Rodrigues Ribeiro, Luiz Felipe Costa Ramos e Maurício Quinhonhas Vega. Parabéns, foi super difícil a gente conseguir chegar nesses 6. Lembrando, então, para os selecionados que eles vão então, apresentar na semana que vem. Cada um vai ter 10 minutos para apresentar, então, por favor, tentem ficar no tempo, tá? Porque nós já temos 6, então, para a gente não passar muito do tempo, façam uma apresentação de 10 minutos e as perguntas ocorrerão, então, no final da sessão. Tá bom? Então, agora eu vou passar para o inglês para a gente poder, então, apresentar o Andrej para vocês. So, now switching to English. Thank you very much, Andrej, for accepting our invitation. It's a really, really pleasure for us. It's an honor to have you here. You'll see a lot of things. Andrej, as Magnum will say, is been working with mass spectrometry to use genomics for a long time, and it's like a really honor for us. Without further ado, I'd like then to give the time for Magnum to present Andrej Shevchenko for us.
Thanks, Thiago. Can you hear me, guys? It's okay?
Yes.
So, good afternoon, everyone, and welcome to another seminar of my friend is MASSA, organized by the Brazilian Society of Proteomics. And today is my great pleasure to introduce Dr. Andrej Shevchenko. And Andrej began his career in 1984 at the Institute for Analytical Instrumentation in St. Petersburg in Russia, where they were doing pioneer working on peptide mass spectrometry. And Andrej then remained there until 1994, when he became a visiting scientist at the European Molecular Biology Laboratory in Heidelberg, together that time with the group of Matthias Mann and then Matthias Wilm. And Shevchenko obtained a staff scientific position at the MBL, where he remained there until 2001, when he joined the Max Planck Institute of Molecular Cell Biology and Genetics that has been recently created in Dresden. And he joined as a group leader, and he was one of the few scientists to have a fixed position beside the directors. And Andrej has several ground-shaking contributions. I'll cite one, that is the paper of mass spectrometry sequencing of proteins from silver-stained polyacrylamide gels. And this paper has more than 10,000 citations. And if you look the authors of those papers, so Andrej Shevchenko, Matthias Wilm, that's now University College of Dublin, Old Worm, that's now in EvoZap, and Matthias Mann, that's the director of Max Planck Institute, and they published that crowd-shake paper in analytical chemistry in 1996. And the same group that was there in this very rich environment, they create other seminal contributions to science. I'll just cite one. That's the creation and application of the tandem affinity purification to characterize protein complex, bringing down the functional data to the MS world. So Andrej has more than 6,000 citations in more than 200 high-profile papers. And it means that Andrej's contributions to science, to science are already an important chapter of the history of MS-based proteomics and lipidomics. And from another point of view, Andrej really knows how to balance personal life with work. He loves skiing and especially being together with his family. And I still remember him with the baby stroller around with Sophia, that probably now is already a young lady. So it means that Andrej has a really beautiful family, and he really cultivates his atmosphere in his lab. And his lab has not only produced great science, but already 10 babies, and Hugo, my son, is one of them. And so with that, Andrej, thanks for accepting the invitation in name of the Brazilian Society of Proteomics, and now it's with you. Thanks.
Thank you so much. Can you hear me, right? The sound is okay, right?
Yes.
So, all right, perfect. Then what I do, I switch to another screen. And I apologize, I cannot go for the presentation mode for some odd reason. So, I mean, it will be a bit, some slides will be a bit screwed up, but I will get it anyway. So, thanks so much for the invitation. Really a pleasure, especially in these times and everything is on Zoom. I mean, you can't see, right, the human faces for months is, difficult. But I mean, let's keep in touch. Let's get in contact. Right. So the title of, today's presentation is kind of, it's a new subject, which we started a couple of years ago only, and only now it's like starting getting to some fruits. We start first low-hanging fruits, and then hopefully we'll get something more interesting and challenging. So, I mean, it's one of the first times I present this in public, so please be considerate. Ask questions if something is unclear, or if I'm fetching too far, and we discuss things. Okay. So what are our interest, in fact? I mean, since maybe like 5 years, I mean, we start looking at metabolism massively, right? And I mean, sorry for this slide, like a design for the undergraduate students, maybe. The metabolism has two important things. Yeah, first, it makes the building blocks and makes an energy, right? It can consume energy, can produce energy, can consume building blocks, and can build new building blocks or remodel things. So, as you know, metabolism is organized in pathways, and pathways are nothing else than successive reactions, successive transformations of substrates into one another, and which again may produce energy, may consume energy, and require certain cofactors, right? But it's, in a sense, it's a kind of a successive transformation of the building blocks, which is used by the animal body. If you go a little bit deeper, once again, I mean, each of those stages is a simple chemical reaction. Well, simple, it's not really simple, it can be, or it's not simple at all if we consider, structural perspective. But anything when we look at this metabolic pathway schemes, right? The rationale is a chemical reaction. So when we have a substrate S, which is the subject of metabolomics, then we have the product P, which again is the subject of metabolomics. And then we have this mysterious E, which is the enzyme, which is studied by proteomics. And I know, I mean, I want to be a little bit more provocative than it's need to be, but the big focus. It's been a big focus on S and on P, on substrates and proteins. But we can quantify those. People do like classical enzymology for maybe 100 years and then can determine the rate constants and the effect of the cofactors and then, yeah. But would you agree with me that we know quite little on the enzymes? For example, a very simple question: how much of the enzyme is there? Right? I mean, simple chemical reaction. We know how much of the, I don't know, glucose. We know how much of the phosphate. We know how much of the glucose phosphate there. What's the enzyme? We know what the enzyme is qualitatively. How much is this? What is the ratio of concentrations between the substrate and the enzyme in the product in a given environment, right? In a given time, in a given place of, given space of the cell or something like this. So why it happens? Because again, I mean, there's I didn't want to make a story very long, but people, including myself, we very often we forgot that mass spec per se is not quantitative. It's a surprising statement, provocative, arrogant, but in fact you would agree. But that mass spec doesn't measure concentrations; it measures some certain signals, right? And those signals are different for different molecules. Even worse, it's different for different molecules in different analysis, different circumstances, right? So we can create the conditions and adjustments and calibrations such that eventually out of those signals, we can produce concentrations. So it turns out that it's much more simpler to produce the relative abundances. When we compare the same molecule, doesn't matter whether it's metabolite, whether it's a protein, whether it's, I don't know, nuclear, anything you want, lipid. We also do quite a bit of lipids, right? Well, we can compare signals coming from the same molecules, the same class of molecules, which are very much alike. In this case, our quantification is better because ionization is the same, quasi, right? It's not exactly the same because of the measurements is the same, technique is the same, and in a lot of things. But what we cannot do is to compare different molecules to each other. Why? Because it requires moles. It requires molar concentrations. If we compare molar abundances, for example, stoichiometry, right? So we have to, if we go for the stoichiometry or whatever, this transformations, we have to determine the absolute molar concentrations of whatever subjects we talk about. It can be proteins or can be whatever, lipids or something like this. So moles is a kind of a chemical rational, chemical foundation of any rational chemical consideration of the biological process, right? It all starts from moles or number of molecules, doesn't matter, right? And then you can sort of the chemistry, the exact chemistry comes. In the biochemistry is becoming less of zoology, I'm sorry to say, but more of the exact science. Yeah, once we know moles. The problem is it's very difficult to do it. Let me just again, it's I apologize for this long introduction, but let me highlight the importance of molar abundances in biochemistry, molar concentrations, right? When we talk about protein complexes, stable protein complexes or stable protein complexes with some transient tessellation subunits, we characterize them by moles, right? We characterize by stoichiometry, 1 to 1 to 2 to 3 to 4, whatever, right? So we determine we define the transient interactors by being sub-stoichiometric. Their stoichiometry doesn't fit the stoichiometry of the complex, right? So talking about protein assemblies and not talking moles is not possible. Kinetics, metabolic flux. That's what we just discussed, right? We want to have kinetics. I mean, look at this simplest equation which everybody knows from undergraduate school. You see the S, that's the you see the E, it is the enzyme concentration, right? So how can we model? How can we do the system biology modeling without knowing the enzyme concentrations? So we go to the pathways. And then I come to this a bit later. Again, pathways is something which is not existing physically, right? It's not like association of a conglomerate of proteins, although it might be, right? But again, there's certain molarations between the enzymes within pathways, and that's what will be the subject of my talk. But I mean, I want to deviate a little bit, and then just focus you on some other aspects of the importance of the molar abundance. Stoichiometry of proteins and assemblies, lipid assemblies, so protein DNA and whatever. How you can compare, let's say, protein abundance and abundance of a DNA in moles. Proteins and lipids, again in moles, right? Because there's nothing else comparable. Last but not least, second to last, is actually the reference values. When you go to the medical doctor, you give blood, for example, for the clinical analysis, they determine, I don't know, 10, 20, 30, depends on the demand, of the values of the concentrations of different molecules in your blood. When you do the same for, let's say, for lipids, or the same for proteins, the medical doctors actually demand that you should determine this concentrations, right? You do ELISA, you do some assays, but the readout is always in moles. That's what the medical doctor can look at this and say, okay, that's too much of it, or it's within acceptable range, or whatever, right? Again, it's all about moles. That's what the language, what the clinical chemistry understands. And I mean, now it's really last but not least, composition of domains, organelles, and right? When we compare proteins which belong to different complexes, or not complexes at all, different pathways, again, to understand how it's built up, we need molar abundances. So in a sense, having the molar abundance is vital for biochemistry, for molecular biology, for clinical chemistry. The only problem is difficult to make, especially when compared when we go to the proteins. So I mean, then another aspect, which and then that's exactly what's our focus is that in fact, to understand development or change in the process, we have to look at metabolism. So basically, metabolic transitions, major metabolic transition, is the cue for development, and let's say for the cell programming, right? So I mean, you understand metabolism, you understand what happens, you understand the nature of the metabolic shift, then you understand what is actually happening within the system, organ or cell. So in fact, that's our main focus. Understanding how the major metabolic shifts occur and what it takes for the organism to actually to reprogram, to change the metabolic state, what it takes on chemical level and molecule level. So one recent work, which I just want to use this for as an example, we have a bit more and then still in the publication process, is work with C. elegans. We collaborated with Timo Kurchali in the lab. He retired now, but I mean, we're still continuing in the same way. So why it's interesting for us? Because C. elegans, in fact, can live life in two ways. So it starts from the embryo, goes to so-called L1 stage. I'll skip this L2 stage and whatever, and then it make a decision. If there's a food and the environment are favorable, C. elegans, I mean the worms, go into reproductive cycle. Yeah, so go to this L3, and then at some point they become reproductive and lay eggs and multiply. However, if the food is not there, the temperature is not there, many factors, so some sort of the signals are present in sense trouble, in effect, metabolic trouble, then it goes to the stage it's called dower. And this dower stage, the worm can live forever. Well, not forever, but very, very, very long. And let's say in a human perspective, about 500 years, whatever. If we just recalculate the timing, and it has extremely low metabolism. Once again, that's important. Low metabolism, nothing really happens. It kind of dwells till the conditions are becoming more favorable, till the food comes, or the environment is less crowded, or the chemical signals will disappear from the environment. Something like this. So there's a major, and then it just in the dower stage, it just dwells and nothing happens with this. Whilst in L3, it reproduces, it actively lives, right? So basically, there are two forms with drastically different metabolism. And the idea was to use proteomics and actually see in which way the worm actually changes metabolism, what it takes. Does it change the abundance of hundreds or whatever thousands of the enzymes? Or just one enzyme, or maybe nothing at all. What's the mechanics behind this? I mean, this is a known question, right? And it's been asked in the field, and actually people knew what's been happening. So in the dower stage, in the right-hand side, I don't know if you can clearly see this. So the metabolism changes through the glycoside shunt. And the TCA cycle is shortened, and in fact, all the resources are going into the building trichalose. So the worm accumulates tons of trichalose, a lot of it, right? It's like becoming really full of trichalose, and can survive, for example, enormous desiccation conditions or low temperatures. So in a normal normal sort of L3 stage, there's no glycolytic exchange, and it goes through a normal TCA cycle. Okay, so that's what happens on the classical biochemistry level. I mean, let's see what it takes. Basically, what do we need to know is the following. We want to quantify all the enzymes which are responsible for the central carbon metabolism, and give them modal abundance. Let's say concentration, although it's a bit odd if we can talk concentrations here. But let's assume that the worm is a kind of a hollow tube, and we take the worm's volume, we quantify the enzyme, and we determine its concentration. It's also very interesting. Are we talking millimoles, micromoles, moles, or whatever, nanomoles? How we are relating to the common substrates, for example, to the trichalose or to ATP or to whatever metabolites we want to do. And of course, I mean, what people do usually is they go for Western blots. Well, if you have enough money, you start buying the kits for antibodies, and there's an assumption that antibody and Western blotting can be quantitative. So unfortunately, the animation doesn't work, but maybe it's even better. You see, I mean, if you just read what's highlighted in red, I mean, this is the common knowledge that antibodies are used because there's no better way, which doesn't mean that they're good, right? They're reliable. Tons of retractions, tons of problems, and hugely expensive. And the antibodies quality varies, and make the Western blot really quantitative is really, really, really difficult. So the question is, if we can do the absolute quantification, not the relative quantification, but the absolute quantification without standards, I mean, without purified proteins, right? Without antibodies, and purely by mass spec. So the way we did it was the following. First thing is we built a proteomics resource. In a sense, we start kind of running the worms in different conditions, qualitatively identifying proteins, a couple of thousand proteins. I mean, this 4,000, that's probably the top 50-60%. I mean, we didn't really dig very deep because we're interested in metabolism, and then we made a library. So basically, what kind of peptides and what kind of proteins are detectable, and what kind of peptides we can match to those proteins. From those, we selected the enzymes from the central carbon metabolism, for example, here from the TCA cycle, and so what kind of peptides are there. And from those, we selected peptides, which we kind of semi-empirically to believe to be like good flies, right? In such that they also don't have major post-translation modifications. There's no methionine, so they're not oxidized. They're not cysteine, they're not oxidized. There are no miscleavages and something like this. That was called, it's a known thing. It's called prototypic peptides. And then we use QCon-cut approach. But we use it with a little bit different way, with the idea, because now the chemical synthesis of this sort of plasmids, right, is rather cheap. And we decided that we will not be synthesizing peptides, but rather we chemically synthesize the gene. We order this, for some money. But then we can have a resource which we can use forever, because we can express it in E. coli, for example, and then we can have all the proteins, all the peptides at once. So basically, we in silico we sort of created a gigantic chimera at about like 300 kilodaltons over here, right? So which contain prototypic peptides, 3, 4, sometime in 5, for all the enzymes which we use. The glycolysis, gluconeogenesis, 23 proteins, and about 30 proteins for TCA and glycosylate shunt. Okay, so that's so far so good, and it's was rather inexpensive. But then we also thought that what we need is some reference. And as the reference, we added to the C terminus a few BSA peptides. And why BSA? because BSA is the only protein we can you can use as you can buy it in the kit, where the company and the Pierce BSA standard, it can guarantee you the concentration. So I mean, we can at any part of the world, you can just take this vial, you can open the vial, and you exactly know the concentration BSA, because it's a standard in a NIST approved chemical, right? And it will be used as a standard for any for protein determinations, for example, for the BCA, whatever, or any other analysis, right? So it kind of, I mean, I hate BSA as such, because I mean, it can be an impurity somewhere. I mean, it can come from the cell media. You have to be careful, but it's the only standard so far we can buy with a reliable known concentration, right? So if it's like 1 milligram per mil, it will be 1 milligram per mil, but not 0.9, not 0.25. So which is kind of avoidable, but it's very difficult. So we went to this. We went this way. Okay. So we put some tags. Twin strap tags, cleavage site, but we never use this, so it's basically a rudiment. Later now we kind of we use some sequences to protect the N terminus, the C terminus, but we don't really use these tags anymore. The most important thing is, so there's a plasmid where the prototypic peptides are concatenated, and there are many of those, and the whole thing is about 300 kilodalton, and it contains a single band expressed protein, massively expressed in E. coli, contains all the standards that we need. I mean, is it really the case? Because I mean, when we talk to, let's say, to people who used to use qconcat, they say, well, I mean, that's one they express this construct as one of the least abundant proteins. I don't know. I mean, that's not our experience. We express them massively. You see, that's let's say this huge band. That's exactly the chimera, which we use. We easily get like 99% sequence coverage. Maybe we don't cover one amino acid because of the some modifications or whatever. And importantly, we did it from gels here. Now we switch to in solutions. Also, the important thing is that you can cut this chimera from the gel and it's pure, right? So there's no like a degradation products, and the ratios between the peptides, I come to this in a minute, are exactly stoichiometric. So we have one standard, and the way how we worked it was the following. Again, we want to do the gels because I mean it's slow, yes, but we want to be very accurate in this case. What do we do? So you see at the bottom here, there's a cell tissue lysate. So we cut it in 6 slices, the big one, I mean the fat one, because we want also to increase a little bit dynamic range. Then we take a chimeric protein. That's the big, it's just here, this small cartoon, but doesn't matter, we take it out. And then we take out the BSA, the known amount BSA, again, the band, and digest them together. What do we do is the following. We use peptides from the known amount of BSA, which is in here in green, to quantify the chimeric protein, where the BSA was included. I forgot to mention, we label this, metabolically label this with the N15C13, exactly like you do Silac or something like this. Right. So basically, we use the known amount of BSA to quantify the chimera, right in the same co-digested mix, right, and in the same LCMSMS, and then we use the known amount of chimera, now with target peptides, to quantify the target protein, all the target proteins which we need, right here and there. Okay. So once again, in one tube, we co-digest BSA. It's a reference protein. We determine the amount of chimera by BSA because they share peptides, and because chimera is the labeled molecule and metabolically labeled molecule, right? And so once we know the amount of chimera, we calculate the amount of any protein which we use, which we want to quantify, and we do it from the gel. We give us quite some flexibility because we can use any amount of SDS. We can use, we can clean this up. We can use the sample preparation. We can concentrate, But again, I mean, it's kind of slow. But we are now we're now doing it also gel free. So interesting thing is the following: that okay, so we can produce some numbers, right? Some quantity, and maybe this quantity is actually reasonable. But I mean, is there any proof that this quantity is actually correct, right? Because you always see peaks, you can correlate them. So we did quite a bit of control, but one is important, and I want to talk about this. It's worth than stopping at this point. So because what we can do, because we don't synthesize peptides. And then we also showed that if we digest this chimera, the digestion is complete, and the yield is almost like 100%, whatever, it's stoichiometric. What you can do, in fact, you can compare relative abundances between the peptides in endogenous protein and in chimera. Well, imagine that we have and use it as a quality check. Well, imagine that we detect 3 peptides, right? So P1, P2, P3. That's coming from the endogenous protein. And then the same peptides, but now labeled, metabolically labeled, are coming from chimera, P1, P2, P3. So if the digestion of the endogenous protein is complete, and the digestion of chimera is complete, then the ratios, the abundance ratios between P1, P2, P3 should be the same. If they are not the same for any reason, for example, because of the post-translation modifications, or because some biased losses, or because of some miscleavages, right? Then this ratio will not be the same, and that's a clear signal there's a problem. And we don't really care where the problem occurs. Either the endogenous protein wasn't cleaved, or the chimera is not cleaved, or both, or we can see that this quantification is not reliable, and we just have to do something else, right? And that's at the right hand side, you see here is a bar diagram. That's what Bsa peptides, right? And you see the relative abundances of those standards are very close to, apart from one, which is miscleaved. But even if we add this whatever miscleaved part back, which is like in red, right? You see that it's reinstated to the expected abundance from the chimera. So it means that we have, for any determination, we have a quality check. And that's what the QConcat and especially also what the peptide synthesis methods are missing, right? They're missing the quality check. So you see that, let's say the molar abundance is calculated for different peptides can vary a factor of 2, factor of 4, can even factor of 10. And then basically, what do you do? Should you average? Should you take the highest? Should you take the lowest? Well, here there's an inbuilt quality check, and yeah, I'll show you in a minute that that's really important. Okay, so that's been the published method a couple of years ago. MS Weston, and then we applied it to quantify those metabolic enzymes in the L3 stage, reproductive stage, and in the DAUR. And then you see at the left-hand side the green and yellow. And I want to talk about this a little bit more, right? So once again, DAUR is a German word for like staying, keeping, Dauer, I mean, long something, It turns out that actually enzyme concentrations in many Dauers are actually higher than the concentrations in the L3 reproductive stage. I mean, the low metabolic stage doesn't mean that the enzymes are degraded, right? Because when you talk, especially like to somebody who is not really doing the metabolic studies like in depth, like occasionally say, yeah, of course, I mean, the enzyme levels should be low. Yeah, but I mean, this is expensive, right? To degrade protein is expensive, and to build it back when the good favorable conditions are coming, and to synthesize, it just takes enormous energy and resources. So basically, when the metabolic transition occurs, nothing really degraded. I mean, the ratios are changing and the stoichiometry is changing in here. So that's interesting. So, and then another thing is, look, there are tons of speculations. What concentrations we are operating? Are we millimoles? And then there's some aggregation and the phase transitions and whatever. Or we are in micromoles, nanomoles. Interestingly, and then for me as a chemist, it's very remarkable that in fact the concentrations of the metabolic enzymes, which are expressed and produced completely independently, are very similar. They are different, maybe a factor of whatever, factor of 2 mostly, right? For many, actually, I mean, if you compare, let's say, the yellows to yellows or green to greens, right? I mean, it's a factor of 3 maximum, and they are the micromolar range, not millimolar, but micromolar range, right? Somehow, I mean, the nature, the animal has means to equalize their concentrations, not to exact level, but kind of close to. Interesting. So what we can do, we can then take metabolic map, like in here, like known undegraded level central carbon metabolism cycle, and then we can add on top the abundances. Each of those are in moles, and you can compare the abundance in L3 and in Dower in the reproductive stage and non-reproductive stage. And also very important, you can compare the enzyme concentrations to other enzymes within the same or within different pathway. You can vastly simplify this scheme by, for example, taking one of the enzymes as a unit, and then normalize it to other enzymes to that. And now once again, that was for me. We published this, but I want to work on this a little bit more and understand what does it mean, right? It looks like they're kind of following the certain like quantums of certain fixed rationales. For example, there are two most important enzymes. You see, 1 and 1.2. But then there's a couple of those which have exactly the same abundance, normalized abundance, like 0.2. And then 2 times more, 0.3, and then 0.7, something like this. So in a sense, there's no continuum within those pathways. When the molar abundance is kind of vary at random, but you see, it is an impression of a very well organized structure, right? So nothing is left at random. There's no like a distribution from anything from 25 to 0.5 or whatever, right? It's a very organized structure when you look at this in the correct way in moles. Okay, so far so good. What we can learn from this? What we can do is now actually look what is the stoichiometric change in the. Just give me a second. We can do this right there. It turns out that we have a branching point in here. So one in the we better look at this here. All right. So unfortunately, no animation, but anyway, right? So we have two branching points, 1 and 2. The branching point number 1 actually determines if the substrate, the carbon, goes into glycosylated shunt over here, right? Through the enzyme isosaccharide lyase, right? Or it goes into the into the long way and uses the isosaccharide dehydrogenase with the CO2 loss and goes through the energy production, right? The same situation is number 2, where it comes back and kind of keeps burning keep producing energy through the Krebs cycle, or it goes to Dower and goes to the production of let's say trihalose, something like this. There are two branching points. And it turns out that although the concentrations of the enzymes has not changed, and even the concentrations in the Dower are increased, the stoichiometry changes such that the enzymes start competing for the substrate. So everything is healthy, nothing is degraded, but only 2 enzymes in here at first branching point get the different stoichiometry, all right, different abundances, such that this the glycosylate shunt is preferred, and it's still the substrate, although the concentration of others, is remain exactly the same. And then when the signal comes, and then we're working on this now, we want to understand how the sort of the back transition from Dower to the reproductive cycle, how it occurs, and if it still has a memory of being a Dower. The enzyme are there, they are ready, and they're waiting. So that's very interesting. So then the same situation is with the second branching point, a twofold difference, not in the abundance, not in the whatever signal ratios or whatever, in the molar ratios, in the concentration ratios, twofold difference, and only twofold, and the whole thing goes into the trihalose synthesis. That's the second, that's number 2 branching point. So in a sense, the major metabolic shifts here, major metabolic transitions are occurred by changing the abundances, the molar abundance of 4 enzymes at 2 branching points. The rest stays the same, and the concentration don't change, right? That's a very kind of rational organization of the cycle. There's no degradation, right? There's no losses. Nothing is lost. Everything is on standby. Okay, so that's what I'm talking now is like a proteomics story. So basically, I will skip biology, especially because, like, for me, it's an episode in a sense, and because we want to do it like more globally on a more organism in a bigger level. But interesting, that's what we noticed is the following. So look at this scheme. You've seen this before with stoichiometry, right? So we know the enzymes. And we can normalize them to one of the enzymes, or we can normalize it to total, whatever we need, because we know moles. When you know molar concentrations, you can do whatever you want. It's completely flexible, right? Because you can compare enzyme to enzyme, you can compare enzyme to different proteins. Everything is comparable. It's transparent. So what we thought is that, in fact, this constellation of numbers is a unique, or maybe unique, kind of identifier, classifier of the metabolic state, right? So kind of a QR code, if you wish. That was actually, we made the slide and we introduced this QR code before this COVID and whatever. All right. So I mean, kind of a QR code, a stamp. And then, since you quantify it in moles, your reproducibility will be, in fact, good and not really dependent on. You don't need batches. You don't need to produce like in a series of the hundreds of the analysis in one batch to be consistent. You can actually, we also check this. We can reproduce those concentrations years after, because again, we have a standard. That's the thing. So it's difficult to reproduce relative changes, but once you have a standard, the internal standard is becoming easy, right? Especially if the standard is produced in situ in a co-digestion. So in a sense, this is a kind of a QR code, and you can compare metabolic states even without knowing what it means, and you can see the transitions and what's been changing, right? What for example, my idea is, I talk about this a lot, but I mean we start moving on this. It looks very promising. For example, we can go for like transplants. Is the good transplant or the bad transplant? Good transplant means it's very similar metabolism. Is it similar metabolism or not? What we can do this? We can actually we can get the QR signature and see. I mean, we can compare signatures to signatures. We can compare. We can use them as a measure of goodness for, let's say, for the tissue or for the transplant. So lots of possibilities. There's no QR coding, and it's very simple to determine because it's just coming. It's coming from a single analysis, and basically whatever. I mean, you have it even if you don't need this. Then, of course, the question which you would ask is, well, I mean, transcriptomics can do the same, apparently, but kind of faster and arguably better. And we all know that the levels between the proteins and transcript don't correlate. It's been known in the field for many years, but maybe they don't correlate globally or poorly correlate globally, but maybe if you go like for a more confined system, there's a better correlation. And in fact, here there was quite a bit of disappointment because what you see how misleading the transcriptomics data can be. I mean, I'm not saying it's not useful. They are useful because they give you coverage, right? But when you start looking in details, the mechanistic details, and here we see that the transcriptomics can reasonably produce the branching point, and the first branching point you can identify as one, but completely miss the second. And these data points here are the same enzymes which you quantified by mass spec, and you see that in many instances the transcript are changing, but there's no change in the protein level, and vice versa. I mean, the transcriptomics give you coverage, give the confidence, but still, misses the branching point. And it misses the way to explain mechanistically how the metabolism actually changed. And it's a very good transcriptome, really high quality data. So it's kind of it's probably inherent limitation of using transcriptomics for this, piecing together the mechanistic details. And especially, please keep in mind that what we are talking about a very small difference, effect of 2 in stoichiometry and effect of maximum 4. A little change. And it's sufficient actually to steal the enzyme and completely block the pathway without actually blocking this, right? The pathway doesn't see the substrate anymore. That's all. All right. So but of course, what you say, it's kind of cool. You can make standards, you quantify it, but you have to decide on the proteins which you want to quantify before doing experiment, right? Which is not very efficient, because if you make a discovery, you see some interesting trends, you want to look at more proteins, and then you have to redesign the whole experiment completely. And of course, it will be very cool actually to know if we can do the absolute quantification in moles in the proteome level. And if you look at the literature, of course you can, definitely. So I mean, there's quite some methods. I mean, one of the most popular is IBAC. It's based on the external calibration of some protein, pre-digested protein standard. There's a proteomics ruler, which uses, I guess, histones right from Matthias Mann. There's a top N or whatever, high 3, like basically considering the abundance of 3 top peptides in a row, and then give you some estimate of the absolute quantity in moles. And we tried this. And the problem is how we see this. That's a fully published data. It's not only data. We see that those methods don't correlate to each other. I mean, maybe just it's okay if you kind of use IBAC consistently, and then you have certain sort of experience, and if your data are biased, then are biased in the same way. But we just took published data. We didn't process them. I mean, the numbers are just taken straight from the Excel spreadsheet. They are calculated to the copy numbers. That's all HeLa proteins, very standard system. And you know, I mean, the correlation is there, right? It's, you know, but it's rather poor. So this sort of this fine analysis, you know, when your interpretation are based on the fact of whatever 2, are hardly possible here, right? Just because, again, I mean, they don't talk to each other. Even if 2 different labs are using the same software and the same basically setup, the correlation is no better, right? I mean, Proteomic Ruler 1 and 2 are 2 different labs, but just, you know, see, the R square is 0.6, 0.7, too low. So what we thought is, why don't we use the same approach as we used for MS Western, but do it like a bit on a global scale, assuming that we have the good, if we consider only good peptides, right? They will be flying more or less in the same way. I mean, we'll want to exclude super good flies on super poor flies and see if there's a median, which will be more informative. And we absolutely need the internal standard in the mixture, because between the experiments, the intensities can change quite a bit. So we made an internal standard composed of good peptides, but what we've done, we scrambled them. I mean, it's in the paper. It just showed up. So we use certain ways of scrambling, you know, such that their flyability and physical properties have not been really strongly perturbed. So we took quite a number of those. So we designed in the plasmid such that within the sequences such they reflect the average properties of the good peptides, protein peptides, you know, and an idea was to use the median intensity. And actually, what's interesting is that we thought if there's any median, or maybe there's a distribution, will always be the same. So we took these plasmids which we had, the constructs which we have, different size, 80 kilodalton, 300 kilodalton, or whatever, but they come to the same median. So I mean, if you look at the if you look at those peptides and look at the intensities, you see that if you consider more and more peptides, when you have more than 20-30 peptides, it arrives to the stable medium. So in a sense, you don't need to have infinite number of good peptides to get this. And then but then of course it's also experiment dependent. That's why the standard has to be added into the reaction system. It has to be co-digested with the means. So what we've done, again, this will be a slide which I just made considered an animation, but anyway, that doesn't work. So the idea is the following: that we design the same standard, the same type of a standard. We used to call it FUGES, for fully unlabeled genetic new standard. So we added BSA, which is in red in here. Below is in gray and green is the native sequence. The up in there it's the scramble sequence. So they are not like not identical to BSA. We disperse them through the peptide backbone of this chimera, and also we took the good peptides, just protein peptides from different sources, and also scrambled them. And we took them many. It's about like 40. So, and then the idea is the following: that we take here's shown with gels, but this works without gels, so also no problem. So what we can do, we can take the material, we can add this whatever fuges standard in there, known concentrations. We quantify the fuges also through the BSA. We add BSA spike in there. So I mean, this is sort of the whole thing. And then what we do, the software publicly available, it just takes the peptides of the fuges peptides, calculates the median, and then use this as median value, single value, as a single point calibrant. And in this way, we can quantify anything which is detectable with very reliably with 3 or more peptides, and the more peptides, of course, the better. And in fact, it outperforms the IBAC, because it's internal calibration, not external calibration, and it also outperforms the top N, because surprisingly, the quantification based on the sort of the best flying peptides is also not reliable, because actually they are best each in a different way. So the difference in the abundance between the best flying peptides is actually quite big. And that's why the estimates based on the best flying peptides also subjected to significant deviation, right? So what we have to do here, we're getting the median peptides. So we compare median from the standard and median from the proteins, and actually we can be within plus minus, I mean within 20% of the accuracy, which is not ideal, not like 10% what we get with the MS Western, but still good enough. I mean, 20% is not a factor of 2, and it's not a factor of whatever x and 5, and it's also very reproducible. All right, so that's actually will be the way to do it in a global scale to whatever is detectable, and based on what I've tried to explain, we make certain conclusions, right? So thing is that what's especially like attractive to me is that if we do the molar concentration of enzymes, right? So we provide a generic resource. This resource, I mean, our estimation can be correct or arguably not correct, but it's comparable. And if any of you, for example, repeat those experiments with C. elegans and look in our data and compare, and you see, okay, so here's a deviation, right? Maybe Andrej did some bad job in here. We can correct this, right? But if you only like compare trends, which are built from relative abundance, like heat maps or whatever kind of bioinformatics you use, right? You will never understood that your data at fault or my data at fault. So we keep producing data without really, which don't really talk to each other. In the way, if I made my data or you make your data available, doesn't make any difference in here. I mean, I can reprocess your data and kind of, have the same result or different result because of the different experiment settings. So my argument is that we have to do exactly what the lipidomics people are doing. I mean, when we are also part of the lipidomics community, is that to make it reproducible when it goes to the disease diagnostics, to like human lives, they have to be comparable. They have to be in moles. So it's generic and expandable for sure. Very odd idea on the QR code. It looks like it's working. We see how far we can push this, especially for complex diseases. Maybe just worm limited, but I mean I love this idea. I love the idea of the QR coding. I mean having a metabolic stamp, which again is built on some transparent rationale, can be compared. All right. So I mean it turns out that metabolic transitions don't really require major metabolic transitions like what's in common between Dauer and reproductive FL3. I actually orchestrated by subtle difference in the enzyme stoichiometry. That's basically about the competition for the substrates. But nothing is degraded and nothing is wasted, right? And the differences again with switching metabolism are rather subtle. So if we're doing proteomics experiment and look for a factor of 10, that's not going to work. There will be proteins which are different by a factor of 10 or factor 100, but the key switches can be extremely low, factor of 2. You've seen this, right? And it's a factor of 2 in the stoichiometry, basically within the same pathway. It's not in the molar abundance. Arguably, so it's a new data, the accurate, I mean, I underscore accurate quantification can be, or reasonably accurate, right, can be done also on a proteome-wide level. But again, as a note of caution, well, we have to use this, right, like systematically and see what's going on. If it's really any better than other methods, and I hope we are, because we have internal standard. All right. So that's a little bit chaotic presentation because of the I had to kind of adjust it to no animation. But anyway, so that's the beautiful work from BrProt and Mukesh. Mukesh now in Harvard, and BrProt is now joining the company. And with assistance of Henrik and other members of the lab, and Andrej, whom Magna might remember. And I want to acknowledge this Sidor Penkov, who helped us a lot with biology of C. elegans from the former Kurchali lab, and of course the protein expression, Alena Bogdanova, who helped us to make those standard proteins clean and in big amounts and big numbers. Thank you so much.
Thank you, Andrej. Amazing.
Yes, so I'll then go back to the. Right.
Yes.
Now
we can see each other.
We can see each other.
So, can I start, Thiago? Maybe I'll start, and then people can start making questions in the chat. So, Andrej, I think that's the way how we want to read if the pathway is active or not active, and the metabolic pathway. And in order to understand that, sometimes the change in concentration of the enzyme is not so important that the change in the proteoform, let's say. So we are adding a phosphorylation that has a biological meaning in one specific peptide, and that will give you the readout that this pathway is active. So how can we deal with that? How big is the challenge of doing that, and how deep we So maybe I can do the other question. So first we can start with this one.
We can start with, I mean, thing is, that's absolutely correct. So, I mean, the minus of this approach, right, is that you don't map the PTM. It can be misleading because then you talk a protein abundance, and abundance doesn't change, and it's regulated by one phosphorylation site, right? And you don't quantify this. So, I mean, this is the fundamental limitation. You can do nothing about this. But what you can notice actually is, that for example, you see that the protein is not changing at all, or changing in a bit different way, and it will be indication that it's regulated by something else, by post-station modifications. So we tried to. There was a few publications how you can make the in E. coli, you can make the phosphorylated proteins. I mean, proteins phosphorylated at particular site. It's kind of re-engineered E. coli with some edit enzymes, but it didn't work in our hands. We could not produce any meaningful peptides. So for phosphopeptides, well, it's still kind of the synthetic peptides is the way to go. Right.
Yes. And
but I mean,
you can sensitize the peptides with the modification and spiking, yes. But then you do have the chimeric, this easy way of production, and so I mean the cost is much higher and so on. Probably
that's not even. I mean, Magnus, not about the costs even. I mean, the costs are important, yes, for sure. The problem is because when we start comparing, quantification by peptides, by synthetic peptides, and what we do by chimera, right? We see that what was the most troubling is that when it goes by peptides, the difference, because sometimes people report the raw data. So the difference, if the protein is quantified by 5 peptides, or 3 peptides, 5 peptides, I mean the difference between peptides can be huge, right? I mean here, for example, I'm unhappy if we quantify with the accuracy of, I don't know, 20%. Well, kind of, yeah, not really nice. But there, right, when you use peptides, I mean, the difference is factor of 5. Factor of 2 is this minimal. It's a very good data. And there is no internal check. Why is this? I don't know. I mean, thing is, first, when the peptides are produced, and then they quantify usually by AAA. And we see that's for some reason, I mean, the AAA can easily give you 20-30% deviation. Right, Another thing is that maybe you can do better, but also the peptides, they live their own life. So they get modified, they disappear somehow, absorption or God knows what, right? And somehow your standards are not really robust on the long term, in the long run. So in a sense, I mean, the beauty of this approach, which we kind of advertise here, is that the standards are produced in situ in this particular moment, right? Right in this tube. And they are stoichiometric, because we look at this very carefully. I mean, because it's a kind of completely random protein, right? And when it gets digested, I mean, it's a random coil, basically. There's no structure. I mean, it gets digested in seconds, basically. Very simple. You know, for example, to digest BSA, you need hours. You have to really to make it complete and whatever. It just take a couple of hours. These guys are digested like with explosion, right? Like in minutes, and it's produced there.
But actually, it has to emulate somehow the digestion process also, no? Or because if that's faster than the normal protein, the endogenous protein process, then you will not be so
exactly. So the trouble can be that so the endogenous protein is not digested, but the chimera is digested, right? Mm-hmm. So that's exactly. So that's why you need this. You need a couple of peptides, right?
Yeah. I think this really smart is ratio between the endogenous one and the chimera of the peptides. It's really smart to eliminate peptides that are not good for quantification and so on.
Exactly. Yeah. So I mean, here's the thing. You know, I mean, we don't really care. You know, why this? I mean, of course, we're interested to learn what's going on, right? But I mean, if there's a disagreement, if you take like the 3-5 peptides, right, and there's a disagreement between the ratios between endogenous and chimera, right? So it means it's a clear message. It's not reliable. Right? Yes. It doesn't pass the quality check. Whether the chimera is not digested in this particular thing, or the endogenous protein is not digested, or the peptide is not modified, right? That's kind of a secondary. But the message is, what if you don't have concordance between the quantification of these 2-3 peptides, or 3-5 peptides, whatever, 3 at minimum, right? Don't do this. Or do it, but kind of know that it in fact is not reliable. So that's why, I mean, I will never accept, let's say, single peptide quantification, which people used to do, because, I mean, what do you want? I mean, you want to synthesize peptides, right? You know, I mean, it's expensive. You have to keep them. You have to validate the standard mixtures. A lot of work, right? I mean, you cannot afford yourself to be 5 peptides. And in fact, you can even if you can, right? It doesn't mean that it will be any good, right? Because in the quality of synthesis is different. Peptides are not stability difference, right? In half a year, you see a completely different picture. And the more peptides you take, I mean, the more sort of confused you will get. So that's why, I mean, this interesting, the beauty, it's kind of underestimated message from this QCon cut approach was actually that you produce the standards in situ now, basically, right? Yes. That's what I mean. Yeah.
So maybe I'll do one more question and then.
Sure. I mean, go on. I have infinite time. It's evening now.
You got really fast through that, but I'd like to know a little bit more about the pipeline to design the peptides. If is there any automation on that? Is there any database search already and validation of good peptides? How do you validate? So this peptide is perfect, it's not good, or so on. I know the ratio, but the other part and so on.
So I mean, there's certain there's a software, but you don't need it. In fact, I mean, there's a in fact, what you should apply the sort of common sense. The best way, of course, the best way, right, if it's possible, is that you make a kind of the preliminary experiment and see what's detectable, right? You can do it blindly, but I wouldn't do this if it's possible, because anyway, quantification experiments takes time anyway. So what you do, you make a preliminary, like a quick and dirty run and see what peptides are detectable. For example, you detect, let's say, 5 peptides. From those 5, you can have a look. Is anything which doesn't have methionine or cysteine? Very simple rules, right? There's no miscleavage, and you can also look at if there's any miscleaved product in there, right? There's certain rules which you should probably avoid, the let's say the acidic amino acid at the end terminus or whatever, right? So in a sense, the common sense, and then also you because for you it doesn't really matter. Make to put like 3 or to put 5. Just put 5. You'll be using 3 for quantification. But I mean, you can afford yourself to be a bit sloppy, because you can get more peptides in there. It's the same cost, right? And see what works. So then we have the software actually to do the MS Western, to pick up those peptides, and it's public, it's downloadable. We can help in here.
And just one detail, for the endogenous protein, do you also collect the signal for the whole gel to integrate the signal of the peptides for the whole gel?
Yes. I mean, if you want to do, you can do this in the single band. I mean, it's like in a closed molecular weight range. For example, your protein is 50, you take, I know, 70 to 30, whatever, because it can be quite big. Right? Or we used to have the full gel. We just cut it in 6 slices. Empirically, more you don't. But also, like you might want to do it a little bit intelligent, because you don't want to overload, right? So if you have antibodies, right, you want to cut antibodies separate and then the rest. Again, I mean, common sense. But now what we do is that we're trying to avoid gels, and then there's still unpublished. I can talk about this, but it will be difficult on words. We use it for, let's say, biofluids, for like CSF, where we don't use gels, and we use like a mixture of shorter chimeras. So this here, like we had chimeras of about like 200 kilodaltons, whatever, 300 kilodaltons. But in fact, let's say for biofluids, I would go for, let's say, 5 together, or 5 short chimeras. They're cheap to make, easy to make, and can do in solution. You know, but there's some tricks involved with this because of these BSA, quasi-BSA peptides. But the concordance of data is amazing, and it's still not published. But we are writing this up. I can send you this preprint as soon as passes the first check.
So, Andrej, I don't know if you can read the question.
Sure. Wonderful presentation. Thank you. I was interested in the comparison between IBAC and proteomic ruler and the relative low correlation. Did you compare also with the TUTMET? Yes, we did. It's in the paper. It's in the paper. I mean, I can, if you just look for my name, like PubMed, just recently, it's called Fuges, you know, and it's publicly accessible. If you have problem to access, just let me know. I'll send you the PDF right away. But it's open source. I mean, open source. It's open access. Can get it. I mean, why for me it was actually a shock. I can tell you this story a little bit to entertain you. So I mean, of course, the people ask how we can do it. You know, are we accurate on the global scale? And I thought, okay, let's just try to reproduce somebody's data, let's say proteomic ruler on IBAC, and see what the correlation is. And then we saw that actually the correlation, our method and to IBAC and proteomic ruler is very poor. Why? Interesting. So then we start looking for correlation between IBAC and proteomic ruler. There's no correlation. Proteomic ruler against the proteomic ruler, no correlation. And also we've been extremely concerned. So because the major concern is that are you talking the same protein or not, right? Because experiments were different, databases were different. You know, maybe you just compare apples and oranges, right? Like semi-comparable. I mean, we look in the sequences, we did homology. We really made sure that we took maybe half of the proteins, but we really made sure that indeed we take the same proteins, right? So why they are not correlating? In a sense, I mean, because I've been in the proteomics for quite a number of years. In fact, it's quite common, because I mean the best any method, non-commercial method, best works in the lab of their developers, right? There are some tricks which you have to kind of probably know. You know, I mean, the way you do calibration and whatever. I mean, but also like maybe even deeper answer would be the following: that the proteomics community, right, they don't really care. You know, I mean, so you would have to admit this, right? They don't really care, because I mean, if the let's say the correlation is poor, well, it's just poor, right? So it be. It's okay for as long as it's research, right? You got some biology, you got some interesting biological outcome, fine. But what is not okay if when you come to, let's say, to the healthcare, basically to the medicine, to reference values, right? That's what MDs work. This is not okay anymore, right? So when you do the discovery, fine. You can do whatever you want. You can do even blind guessing. Doesn't matter for as long as you pointed to the correct protein, right? But it's not okay when you start talking about reference values, right? And this lack of concordance, it really hits the field and make the sort of the practical medicine skeptic.
And also to share the data, because if you share concentration, so for sure, I mean, you can recalibrate and everything afterwards. Yeah.
Right. So I mean, in the sense, that's why you talk to proteomics people. I mean, you guys can talk to yourself, and then everybody kind of, the opinion will be, you know what, we are super accurate when you do relative. We can do, I don't know, 5% difference even lower. We can do super low abundance, and when you do the absquant, I mean, this is the very sloppy field. We don't do this, right, in a sense. I mean, talk to your colleagues, and probably the feedback will be the same. So we want to change this. I mean, it's a process. It doesn't. I have no delusion that, like, if I publish yet another paper and then the field changes, it doesn't. But I mean, somebody has to start with this, right? And why don't we just start and see what happens? And maybe like in some years we'll be completely forgotten and something else will come, and I will be gladly using this. But I mean, we have to start. Are there some challenging moving the approach from gel-based into liquid digestion? Yes. And actually, it was very surprising. It's not because of the in-gel digestion is not complete. It is. It's all cool. I mean, the thing is that what we noticed is that we were lucky when we start doing this MS Western, that we start cutting bands of the chimeric standard from the gel. And we thought, okay, so we do it because of the consistency. But in fact, what we've done, we purified this protein. See, we thought, okay, so we want to have gels against gels, like for ease of sample preparation, membrane proteins, whatever, because some of those proteins which I showed you, quite a bit of these are membrane proteins, right? But in fact, the plus which we haven't realized was that actually we've been purifying the chimera. When we cut the band, you purify this effectively, because I mean when it's overexpressed, there are quite a number of proteins which are of this chimera, which is not complete, which doesn't go straight to from the N terminus to the C terminus, right? It's like a fragment, and it might and the ratio, or let's say the abundance of those incompletely expressed chimeras is more on the N terminus than the C terminus, right? Because translation comes from N to C. Right. So, and then once we purify this, we have a clean chimera, perfect stoichiometric ratios. When we take the whole soup, it doesn't really. People say, okay, so because of the protein background, it's not important, really. I mean, we have tons of this protein, and you have to work really hard to see any E. coli proteins there. It's really nothing, percent. Well, the most important thing is that it's incompletely translated proteins, fragments which are floating in there. And that's the only problem which prevents us from moving with like heavy chimeras, whatever 200-300 kilodaltons to the insolution. That's the only thing. So that's why what we start doing now. And then said to me, if somebody's interested, ask me, send me email. I'll send the printout. That we start doing this with shorter peptides, shorter chimeras, 40-50 kilodaltons. And we used to use a way to balance them, and we put a mixture like in the soup, and it works as good as the long chimera. So if you want to do it fast, and then you might want to do like multiple chimeras and have some balancing peptides included. I mean, read the paper; will give you the full details. There's no secret. The idea is very simple. I hope that answers the question, right? Yeah, sure. So once again, I mean, when we start kind of applying this systematically, like day to day, right? We learn quite a bit why this works and why it doesn't, And we thought it's mainly because of incomplete digestion, sample preparation. No, I mean the biggest problem is incomplete translation of the gene. Oops.
So, Andrej, there were more questions here.
Sure.
Yeah, I think they were more or less the same, because there was one for Fabio that he was asking how to measure activated and inactivated enzymes. So it's more or less what Magno asked. Also, Janeiro asked the same. So I would like to hear more from you, like more a comment, because for us as mass spectrometrists, guys, we rely a lot on our data, and it's really hard to change, like the researcher's mind, the guys that works more like with the classical biochemistry. So let's say Western blot, as you mentioned. So people really rely on that, and I really don't know why. And as you mentioned really well, and they are aware about that. So sometimes you need to do several assays to save fuel. Also, honestly, I mean, I do, of course, a lot of Western blots because people ask that, but I really don't, it's not reliable. So first, as you mentioned, sometimes it's not reproducible. It relies a lot of antibody, and some we know that they don't work well. And when you use it and you have your spot there, it doesn't mean that your protein is there, right? It could be another form. It could be any other thing. And anyway, what else we need to do? Because, I mean, us from our field, we're already trying to do that for more than 10 years or even 15, since like MRM, SRM. And I mean, what else do we need to do to make people rely on this data that we produce and say, well, listen, Western blot, no one should use it anymore because it's not reliable. What do you think?
My answer is absolutely, I fully agree with you. I mean, it's just, yeah, perfectly. I mean, I can sign anything what you just said, right? My answer would be the following. My answer would be a bit philosophical. What do? I would say just live, do the stuff and see, hoping for the best. Because I mean, what's the history? I mean, the history doesn't care about the human life, right? It doesn't care about Andrej Shevchenko, whatever. It's like X years and we are gone, right? Anyway, right? So in a sense, the good data win. So I mean, you cannot beat by arguments or by like shit throwing, and there can be tons of reasons why Western blots are not working, and there will still be people who are like believing that the Western blot is the way to go because they've been doing this for another 50 years and whatever, right? You can do nothing about this. Just to publishing data and kind of winning by the data quality and findings, right? And the the outdated methods will just die by itself. It's not very rewarding, but that's just the life is, right? Because I mean, you cannot argue with this. And then also like at some point the standardization will come, and then look at the proteomics field. I mean, when we started, let's say 30 years ago, whatever, it was very qualitative. We're just happy to sequence a band. All right, cool. Now, if you do proteomics, which is not quantitative, at least on the relative scale, I mean, something is wrong with you, right? You do the label-free anyway. I mean, but I even remember that maybe 10 years ago, whatever. I mean, label-free, well, that's for freaks. We do Silac, we do whatever, iTrack, we do anything, but I mean, label-free, no way, no, right? Well, now who's doing Silac these days? Very little, right? Because you can do label-free and it's cool. It's actually working, right? So same thing. Same thing is I wouldn't be surprised, if let's say in whatever 10 years, and whoever doesn't do the absquant is a freak. How comes that you do proteomics and you don't do the abs quantification? No way. I don't accept this, right? It's kind of, I mean, it's in a sense it's a human life problem, right? Because our career is short, 20 years or 30 years plus minus before we get to understand what's going, right? And it's gone, right? But let's say for the community, 20 years is just nothing, right? It's like yesterday, right? Sorry for this, bit philosophical thing, but that's exactly the situation is, right? Just keep doing stuff and that's it. Nothing you can do about this. Nothing you can do like to change the community's opinion overnight. Doesn't help.
Yeah.
Oh, no, another curiosity, right? I mean, you try to correlate the concentration of enzymes and their substrates products in the same set to determine enzymatic activity. No, we cannot determine activity per se by some whatever substrates. Just we didn't have the skills. But yes, we have the concentrations of enzymes, right? And that it's very clear that the concentrations of substrates, right? And that's very interesting, because I mean, even if you assume the Km value is about like 10 micromolars or whatever, there are different scenarios where there are more substrate and less substrate, right? But you only learn this when you know the concentration of the enzyme, right? Yes, we did. I mean, it's a simple metabolites. You can do metabolomics, you can quantify them, standards available, straightforward. Oh, kind of straightforward. I mean, it's work, but it's not a Nobel Prize thing.
So, Andrej, just one comment. Another thing that I find really interesting, that in the daughter lines in C. elegans, so you mean that you have the whole machinery there, but somehow the pathway is not active, but it's just waiting to one signal to start.
Precisely.
It's interesting that why do you keep the proteins there? What's the sense of keeping the proteins if you just could? I don't know. I was just thinking. I don't know.
My gut feeling, I mean, what I like about this, and actually it's not really super new in the field, because when we publish this, people say, yeah, okay, this is good. I mean, we thought this way, but now we have a direct confirmation. Is that the protein degradation is an expensive process, and building a protein is even more expensive in terms of the energy, in terms of the amino.
Yes.
Exactly, right? So in a sense, I mean, philosophically, the system, whatever the system is, worm, fly, whatever, it just tries to avoid the energy expenditure, right? Because it's easy to let's say to digest this, the kind of ubiquitinate, throw into proteasome and it's gone, right? But to build it back, and if you have to build the pathway, right, and you have to build, let's say, 5, 6 enzymes, come on, that's difficult, and also very slow, right? So that's why worms, I mean, they transit from, let's say, from dowers to reproductive stage back very quickly. I mean, it's not hours, but like days, right? And then they're reproductive, right? I mean, they can, lay eggs and whatever. So basically, it's more logical and more energy sort of consuming and resource consuming, actually, to keep the system that you need in the future. You may or may not need it. Maybe the worm dies. Never had a chance to see the enough food, right? But it's to keep it ready, and once it's ready, it's ready. There's nothing to do. Everything is ready, everything is floating, everything is correctly folded, everything is there, packed, waiting. I mean, for me as a chemist, it was super good, it was super interesting, because all time you kind of talk to really non-professional and say, yeah, I mean, it's completely degraded, I mean, it's inactivated. Yeah, maybe they are there, but they are not really active. Come on, that's not true. Everything is there in the same stoichiometric ratios.
Perfect. So, Thiago?
Yeah, I think I have no more questions. I think no more from the audience. We just need to thank you a lot, Andrej. It was really.
I mean, my pleasure. Sorry for being this mess with the presentation. I don't know why. Somehow maybe something is wrong with my computer, but I hope you've seen this. I mean, there's only a few slides.
Sure.
The message was like very clearly set for you. So no.
If you guys are interested, send me emails, and we can have a separate Zoom, for example. I'll send you the link, and then we can just talk details. Or I can give you this whatever papers and because I mean it doesn't really have sense to talk big audience and concentrations and whether 37 or 35 or whatever. You take 12 nanograms of trypsin or 10, that's who is interested. We can talk anytime. Zoom is working. I'll send you the link and
maybe I can do one last question. Sorry, Andrej. Yeah, how long does it take if I want to do metabolic quantification? One specific pathway that I want to look for. How long does it take for getting the whole molecular concentration of the pathway?
I mean, the measurement is straightforward. I mean, to make a standard, it's basically, let's put this way. I mean, the shortest 2 weeks, the longest a month, because they really now have this automated, and there's not a big deal at all. Yeah, at all. So I mean, thing is that now it's more problematic to get peptides, because I mean, peptides are cheap, right? But nobody cares about the quality. I mean, you can get amazingly cheap peptide synthesis, right? But then what do? You put it on mass spec, or you put whatever you put it on CMS, whatever. It's one peak, right? But how do you know concentration?
Yeah.
I mean, it's correct peptide, but how much salt is there, right, or water?
You need amino acid analysis, so it's really expensive. So the price of one peptide will be 2,000, 3,000 if you really want absolute quantifier, then precisely with the amino acid analysis and so on. Yeah.
That's what I mean. I mean, the beauty when I realized that in fact it's when we do this chimera, right? You do it once, right, in a lifetime, and I mean, look, it's a very interesting thing. So I mean, you express it in 1 ml of the E. coli, right? I mean, you just really 1 ml. I mean, we don't talk liters and whatever. No biochemical purification, nothing, right? It's just 1 ml. So this 1 ml, that's for you, for your kids, and for grandchildren. Because basically, in fact, your challenge is not to concentrate, but rather to dilute. Because you are measuring like picomoles, low picomoles, femtomoles, whatever, and they are like grams of this shit, right? And then you have to get the proper dilution. We even don't. So far we even haven't solubilized the inclusion bodies. No need to. So we take this whatever supernated and dilute it back to 100. Because there's so much of this. Tons of stuff, right? I mean, and then of course, and after that, I mean, peptide synthesis. I mean, I'm not interested in this anymore, unless it's modified or something like this, a special case. Yes. But for pathway, you can put like 3-5 whatever peptides per protein. You can have like 10, 30 proteins, in one chimera, and then you use it forever. Yes. Yep.
Perfect, right. Andrej. Thank you again.
Thank you so much.
In the name of the Brazilian Society of Mass Spectrometry.
Good. Nice to see you. Yeah. Bye.
Thank you.
We have to say thanks to exposure, nova malícia. So thanks, Andrej.
Yeah. Best greetings to Livia.
Thanks, Thiago.
Bye-bye.